Good morning, everybody. And I would like first to thank the organizer for organizing this exciting conference. And I will present you the ongoing work that I am doing on the spontaneous usage of ultrasonic vocalizations in rodents. And so it's really ongoing. So it's basically sharing methodology and approaches and getting any feedback. So I am interested in involving my studies as a type of personality, so at the individual level. And personality traits have been studied in basic science, for instance, with naked mole rats, and people have studied which individual would be the most likely to disperse, or in species conservation to see which individual will have a better chance to survive to a translocation. In animal welfare, also with cows and horses, to see the most resistant animal to stress, and also specific needs in the different animals, and also in rodent model of disorders, such as, for instance, predisposition to psychiatric disorders that can be translated to rodent animals. And personality traits have been defined as there are 5 of them. So you have boldness, so propensity to react to potentially threatening situation. You have the activity, so the of the animal, the exploration, how it explores the environment and gather information in the environment, and also sociability and the aggressiveness with agonistic behaviors. And these traits should be consistent across time and context to be considered as personality traits. And what I am interested in, so it's also to link personality traits with other abilities, such as problem-solving abilities. And this has been shown in wild rats already, that more exploratory individuals solve problems faster and solve more problems. And also in mice, for instance, risky behavior have been put in parallel with cognitive flexibility. So I would like to apply this type of approach at the individual level in rodent model of autism, with or without intellectual disability. And for that purpose, I need to explore really the behavioral repertoire of the animals, and I am trying to get the most relevant behaviors that can be measured automatically according to the different traits that I'm trying to establish. So, for instance, activity, you can represent it by measuring the distance traveled of the animal by the animals, or the stop and the move events. The exploration can be quantified by rearing and the initial exploration of an environment. Boldness can be measured by measuring the reaction of the animal to a mild stress applied to the testing cage, for instance. And I also put a deep focus on sociability, given my interest for models of autism. And for instance, this can be distinguished in very different sociability models, I would say, with the contact initiation, in which you can quantify the occurrence of approach behavior, so to initiate a contact, the maintenance of contact with the duration of each type of contact and the mean duration of each event. So different types of contact with a proportion of the whole time spent in contact, for instance, in the different specific contact, like nose-nose, nose-to-end, genital, and side-side. The avoidance of contact also with the escape behaviors that can be quantified, and the dynamic contact events, such as the follow and the chasing events that can last for several seconds. And also the passive behavior in the social interactions, so tolerating the other individual exploring themselves. And for the aggressiveness, it's basically the same type of behavior, so initiation, the avoidance, and the social dynamic. But this will be with modulators, such as a different speed level at a proxy for this aggressivity. Or what I am trying to do also is to distinguish behaviors according to the synchronous ultrasonic vocalization. And this is, for instance, in rats, in which you have this 50 kilohertz more positive calls and the 22 kilohertz calls, which are more negative. And so I try to establish the repertoire like this, and we are using it in mice and in rats, so it can be applied in mice and rats. And the setup, the recording setup, is the live mouse tracker system that has been developed by Fabrice de Chaumont, and automatically monitors the behaviors of animals living in groups for days and nights, so as long as you wish. And so you have this repertoire that is automatically gathered. It can be synchronized also with ultrasonic vocalizations recordings that we are recording through Abisoft, but you can use also other types of systems. So LLT is completely open. And we analyze it using USV toolbox, but it can also be analyzed with other systems, and you synchronize this and you get the context of emission of vocalizations. And this is what I'm using to try to distinguish between positive, negative social interactions and so on. So this is the illustration for mice, but we applied this to rats. The system, the live mouse tracker, has been adapted to rats by Fabrice Le Chaumont, who is also on the call. So if you have questions, don't hesitate. And the system, the test cage is a bit bigger, but really the way of working is the same thing for the live rat tracker as the live mouse tracker. And so as a first trial, so I tried to see whether we can combine vocalizations and behavior monitoring. And I tried to do this in a kind of a pilot study in which I used only pairs of animals because I still don't know who is vocalizing. I don't have the nice systems that were presented today. And so I compared the long-event strains with the Sprague-Dawley strains by looking at pairs of adult male rats, and I recorded them for 2 days and 2 nights and try to see which type of behavior I can monitor and how I can refine that. And so, for instance, so we measured the total distance traveled to have a proxy for activity, and we found that the long-event animals were more active than the Sprague-Dawley. As expected. And for the social behaviors, so we found that the total time spent in contact in general was not different between the 2 strains, but we found that the long-event animals have more nose-nose, nose-to-genital, and more side-side time spent in contact than the Sprague-Dawley, meaning that they were more specific in the type of contact. And on top of that, we measured ultrasonic vocalizations, and I focused on sequences of ultrasonic vocalization, not single calls. And we got sequences of 22 kilohertz spontaneously, sequences of 50 kilohertz calls, and also mixed sequences of 22 and 50 together. I don't know if you see my mouse. And also mixed sequences of vocalizations that were going up to 80 kilohertz and starting from 30 kilohertz. And to compare this, so first the quantification of the USV sequences, we found that there was a slight increase in the emission in long-event animals. And the long-event strain was emitting more of these mixed sequences, so these 22 and 50 kilohertz calls compared to the Sprague-Dawley. And which is kind of ambiguous, but I would like to dig into the context of these sequences. And to look at the context of these sequences, so we take each behavioral event and we looked whether it was accompanied or not by ultrasonic vocalizations. And we found that there was a major differences in the strain for this long chase event, where the animals are following each other for a long time and combining with stop and move and so on. And these long chase events were more accompanied by vocalizations in long event animals compared to Sprague-Dawley. And for other things, there were no major differences between the 2 strains. And we did also the reverse analysis, in which we consider each USV sequences and we checked to which behaviors they were synchronous. And we found, so for both strains, so you have a graph of 2 strains and you have the proportion of the USV sequences that were accompanying the different types of behaviors. And what you can see is that the Sprague-Dawley animals here on the right have very few 22 kilohertz calls, these green sequences that you cannot see them a lot. And also in both strains, both of the 50 kilohertz vocalizations, so this red and orange, were mostly emitted during this approach behavior in both strains. And we need to refine a bit this analysis to get a bit deeper into this spontaneous usage of vocalizations. So this would be used to refine the behavioral repertoire. And what we are doing with this behavioral repertoire, we are trying to examine the individual within its own group. So within the group, we see what is the classical trait of the animal, because we know that each animal is specific in its group and that it's also shaped by the group. And so to examine the individual within the group, we are doing a kind of a behavioral profile for each individual related to the mean of the group. So basically, I took the raw data for each behavioral variable that I get, and I normalize it according to the mean behavior of the whole group. So you have the mean here with this dark circle here. And if the behavior is outside of this dark cycle, it's more expressed in the individuals than in the mean of the group. And when it's inside the circle, it's less expressed in the individuals than in the group. And you have this type of typical profiles for each individual within the group. And this is an example that I did with a group of 4 rats, 4 female rats. And you can see here, for instance, the rat 1 was middle active in blue here, but it has a high social tolerance. So it was highly explored by the other individuals, but it avoided also a bit the others in yellow, for instance. And this was different from the rat 4, for instance, which was highly active here in blue, but it has a low social tolerance and also low social maintenance in orange here, and also low contact initiation. So basically this rat 4 was mostly asocial. So this is the type of description that I get now, and I would like to refine this with the USV monitoring. And the next step now is to explore the stability of these individual traits over time by doing longitudinal following of the individual to see whether it's stable also, and also to see whether it's stable across context, so whether it's stable with new behavioral tasks such as foraging, and also to include cognitive tasks that is now possible with the modular approach of the live mouse tracker that Fabrice is developing under the code name Nicecraft. And I also would like to refine the behavioral profile with this USV monitoring, and I will need to introduce this down source detection. And so this is the next step, to see who is emitting vocalization and to be able to follow groups of animals and not only pairs. And also I will constitute a baseline data set with the 2 strains, and both sex is per strain, but I face the question about how many individuals, since we are considering groups, and how many groups per strains. And the same question arises when we apply this to rodent models of autism. What is the number of groups that we need to test, and how is the group composed? Do we put, how many mutant animals do we put, and so on? So this is ongoing questions. And now I would like to spend some time also to present us the side project, so the database MouseTube, which was developed by Nicolas Torquet when I was in the lab of Thomas Bourgeon at the Institut Pasteur in France. And so this database was designed to share raw data of ultrasonic vocalizations, as Kuta presented this morning. And so we share the raw data, but also metadata to be able to reuse the different files. And also we provide a catalog, kind of a software catalog for recording detection and analysis solution. And we also provide examples of protocol, because they were attached to the raw data files. And this was how it was created. And we got a lot of user accounts that were created. Basically, it's almost 1 per week. But we get very few contributors until now. And so the question that we were asking with Nicola was how to make it more useful for the community and to make it evolve a bit instead of letting it die. And so we also increased our team with Emma Cahill from Bristol University and with Laurent Bourry to develop the second version of MouseTube. And we basically kept the same framework for MouseTube, with the raw data that are shared and metadata, but we make the metadata more simple to make the way easier for user to upload their data. We still kept the catalog of software and the examples of protocol, but what we added is a kind of an automatic link with a public repository like Zenodo. When you upload your data, it's automatically uploaded and stored on Zenodo, so you don't need to get to be concerned about the storage. It's done for you because this is one of the problems I could have pointed this morning. We also extended the mouse tube database to other species, and we will have rats and to the help with from Emma, and we also extended to African strip mouse with the help of Elodie Ey that you have heard yesterday. And also we included a reference data set providing data to help people challenge their own detection applications. And so this was constituted from 2 data sets, so one for mice, one for rats. And for the moment, you have a dozen of files from each species in each data set. And these files are more or less long with very different conditions of recordings and with different background noise conditions also. And they were all manually annotated by experimenter. This is what you can see in black and gray here. And we also used automatic detection analysis to analyze each files. And this is labeled with the different colors here. So you have different solutions, but it can be increased and so on. And I used only basic parameters for this application. So maybe they are not optimized. And if you are willing to contribute, we are really happy to get you on. And for each of this detection analysis solution, we quantify, for instance, the number of vocalizations that were detected for the whole of the calls together, and also divided by the duration of the calls, because the detection accuracy can depend on the duration of the calls. And also for each file of the data sets, we provide a comparison between the manual and the automatic solution for each software, by, for instance, measuring the false negatives. So basically the calls that were really actually calls, but were not detected automatically. The number of false positives, so the number of signals that were labeled as call, but that were not labeled manually. And also a kind of an estimation of the proportion of the duration of each signal that has been accurately detected automatically. So this is just to provide some quantification on the different files that we provide. And all this data, so this is a manual annotation and also different automatic solutions are provided as text files. You can also download it and so on. And if you are willing to contribute with your own application, you are really welcome to do that. And to show you an example of what kind of things can be done with MouseTube, we are currently setting up with Elodie Ey kind of a cross-species analysis on the vocal diversity in rodents. And we would like to compare this vocal diversity according to, and to check whether it is related to the species, the social system of each species, or the recording conditions. And for that purpose, we will focus on the male-female interaction context that can be found in all species. And we would need 6 different encounters or individual per species with recordings of 1 to 10 minutes to have a kind of a good overview of the diversity of the vocalizations that were emitted. And for the moment, we got vocalizations, so recordings from lab mice, African strip mice, common hamster, but we were looking for other species, such as the rats or any other type of species. And if you are interested, you are really welcome to contact us. You can see the chat. Leo is putting a link to a place where you can put your contact details, and we will contact you back. And so this is all for now. I would like to acknowledge my different collaborators and also the USVs that is hosting MouseTube. So MouseTube, for the moment, you have only a temporary version of MouseTube, and the next version, so the V2 that I presented, will be published soon, next year, I guess. And so if you have questions about this, you can contact us at the MouseTube email address. And if you have questions about Live Mouse Tracker or Live Rat Tracker, please feel free to contact Fabrice also. He would be very happy to answer any questions. And I thank you for your attention.